Welcome to Bio-Symposia 2021. I hope everyone is physically and mentally well. Is everyone ready to be enlightened about bioinformatics? Because I'm ready. I'm Elijah Rivera.
And I'm Diane Shonka, and we are now officially starting the webinar on Bioinformatics for Novice Scientists.
SIHAY NBO is a national inter-high school biology congress organized by UP Cell Biological Society. This year's SIHAY zooms into the world of bioinformatics, decoding archives, unraveling the biological code through molecular data acquisition and analysis. With the advent of technological advancements that urge to unravel the closed-book science, bioinformatics proves to be a principal tool in collecting the essential codes to grant access towards the bigger world of biology. As we witness the hybridization of the biology with other established fields in decoding the codes of life, let us look forward to bridging the gap between science and technology in evidence-based and precision studies through bioinformatics.
SIHAY NBO is comprised of several contests and sub-events, one of which is Bio-Symposia. Bio-Symposia aims to present recent advancements in biology and biological instruction to students and educators in the hope of using this knowledge to innovate their learning and teaching styles and methods for the advancement of biological education. There will be 2 webinars that will be broadcasted on April 25 and on May 1, having one esteemed speaker for each webinar. Are you guys curious on who these acclaimed speakers are?
The organization has invited 36 of junior and senior high school students from the whole country to partake in numerous biology-related workshops and activities, namely the Science Feature Writing Contest, Digital Poster Making Contest, Junior Life Sciences Research Olympiad, and the Biology Quiz Olympiad, which certainly all have their respective prizes. Each contest has a designated monetary prize for the second runner-up, first runner-up, and the champion for that event. Your school can even win as much as 15,000 pesos. How exhilarating is that? Not to mention, the overall champion from all the contests shall receive a cash prize of 5,000 pesos. Before we proceed with the event proper, we would like to encourage our attendees and viewers to share your thoughts and use our official hashtags, #Biosymposia, #BeBioinformed in your social media accounts when sharing our event.
We would also like to invite our attendees and viewers to please like, share, and follow UP Cell Biological Society's official social media accounts on Facebook @UPCells, Twitter @UPCells, and Instagram @TheUPCells. You may also want to follow us on our SIHAY NBO Facebook and Twitter page @SIHAY NBO to keep yourselves updated on the National Biology Olympiad.
Soon, we will find ourselves in a vast horizon of knowledge. So sit back, relax, and listen, and prepare to learn and gain new perspective about bioinformatics.
Before we begin, please be heedful of the following house rules. Proper etiquette expected among all participants. Any form of discourtesy shall not be tolerated. Kindly key in your questions for the speaker in the Zoom meeting's chat box or in Facebook Live's comment section. These questions will be noted by the organization and will be addressed in our post-webinar publication. Additional questions for the speaker shall be entertained through email. And lastly, to acquire e-certificates, attendance on the second webinar is also required, as 2 codes will be asked on the post-webinar form. The first code shall be announced at the latter part of this webinar.
But of course, this seminar will not be possible without our sponsors and partners. Hence, we would like to acknowledge them for making this event possible. In cooperation with UPLB Computer Science Society,
UP League of Agricultural Biotechnology Students,
Don Honorio Ventura State University Biological Society,
Pampanga State Agricultural University Biological Society,
Biological Society, Laguna State Polytechnic University, Santa Cruz Campus,
PLM Society for Biological Sciences,
Bicol University Symbiosis,
League of Biology Enthusiasts, with Media Associates, UP Society of Management and Economics Students,
UP Biological Sciences Society,
Philippine Society of Cell Biology,
Biology Program Council. Special thanks to UPLB Development Communicator Society,
Symbiosis, the UPLB Biological Society,
and Agham Youth UP Diliman.
Again, we would like to remind everyone of the following house rules. Proper etiquette is expected among all participants. Any form of discourtesy shall not be tolerated. Kindly key in your questions for the speaker in the Zoom meeting's chat box or in Facebook Live's comment section. These questions will be noted by the organization and will be addressed on our post-webinar publication. Additional questions for the speaker shall be entertained through email. And lastly, to acquire e-certificates, attendance on the second webinar is also required, as 2 codes will be asked in the post-webinar form. The first code shall be announced at the latter part of this webinar.
We would like to encourage our attendees and viewers to share your thoughts and use our official hashtags, #Bio-Symposia, #BeBioinform in your social media accounts when sharing our event.
We would also like to invite our attendees and viewers to please like, share, and follow UP Cell Biological Society's official social media accounts on Facebook at UPCells, Twitter at UPCells, and on Instagram at the UPCells. You may also want to follow us on our SIHAY NBO Facebook and Twitter page at SIHAY NBO to keep yourselves updated on the National Biology Olympiad. To formally start our webinar for today, we would like to invite the incumbent president of the UP Cell Biological Society, Mr. Bruce John S. Estilo, to convey his opening remarks.
Good day. I am Bruce John S. Estilo, and I am currently the president of the UP Cell Biological Society, or UPCells. I hope that everyone is well and in the best mind and spirit. SIHAY National Biology Olympiad has been the flagship event of UPCells for 22 years already. This year, we chose to overcome the difficulties to continue to uplift biology and provide learning opportunities to the youth. So SIHAY NBO 2021 was initiated as SIHAY Plugged. Through this, we can communicate biological knowledge to the community. We aspire to foster new friendships through friendly competitions. And we work towards developing awareness and enthusiasm in the field of life sciences. For the first highlight of SIHAY NBO 2021, which is Bio-Symposia, may you, our audience, find learnings and inspiration that you can wield further to develop yourselves. As much as we have discovered satisfaction from the preparation up to the execution of this long week event. While we are not able to see and meet each other personally, may our truest aspirations transcend the virtual boundaries. I wish everyone the most fulfilling and fruitful experience in SIHAY Plug from our screens to yours. Thank you very much and have a great week.
Thank you, Mr. Estilo. Now let us move on to the much anticipated seminar proper. Diane, can you introduce our speaker to our audience?
Our speaker for today will be discussing Bioinformatics for a Better Philippines. He obtained his bachelor's degree in molecular biology and biotechnology at UP Diliman and graduated as a cum laude and was a Department of Science and Technology scholar. In addition to his bachelor's degree, he has a master's degree in molecular biology and biotechnology from UP Diliman, as well as another master's degree in computational biology from Washington University in St. Louis. He received multitudinous awards, namely the Department of Science and Technology Award for Academic Excellence in Science, Gawad Chancellor para sa pinakamahusay na estudyante, undergraduate category, Science and Technology Cluster, and the Gloria Macapagal Arroyo Presidential Award in Science and Technology. Presently, he is a Senior Bioinformatics Specialist at the Philippine Genome Center. We are pleased to welcome our guest speaker for today, Sir Carlo Lapid.
Thank you very much for having me. It's an honor to be here. So my talk is about bioinformatics for a better Philippines. And as you can see, I really just cribbed the title of what's basically the logo of the Philippine Genome Center, Genomics for a Better Philippines. The Philippine Genome Center, of course, is where I work. It is an institution that is geared towards developing a deeper understanding and judicious application of advanced knowledge and emerging technologies in genomics and bioinformatics in fields such as health and medicine, agriculture, biodiversity, forensics and ethnicity, industry, and the environment for the benefit of Filipinos and the rest of humanity. This is a quote from our mandate, our founding documents. And so this is the vision which drives all the work that we do at the Philippine Genome Center. So I thought that it would be a fairly apt title, and I hope perhaps an inspiring vision for the young and aspiring scientists out there. So I work specifically for the core facility for bioinformatics at the Philippine Genome Center. Now, its objectives are to help the country's scientists and researchers and experts in fields such as genomics and bioinformatics. We want to provide access to tools, equipment, resources that they need for genomic and bioinformatic research and advance education in these fields, bioinformatics and computational biology, advance the expertise within the Filipino research community and among young students such as yourselves, because this is still a fairly fledgling field in the country that we want to advance. And of course, we want to pursue research in genetic and genomic data analysis in line with the other research thrusts of the Philippine Genome Center. Among the core team, I want to acknowledge, of course, our director, Dr. Jan Michael Yap, as well as some of my immediate co-workers and team members, Francis Tablizo, Elkin Morado, and Joshua Dizon. We come from different backgrounds. Some of us are from molecular biology, from computer science, from physics, from agricultural biotechnology, but we come together to this facility to do research and work in bioinformatics. Before I talk about uh, bioinformatics as a whole, I want to talk a bit more about what our facility does and our resources. So we provide access to powerful high performance computing servers that are for use for our clients to be able to engage in the research. Just to give you some of the specs, no, these are 512 cores, 2.6 terabytes RAM, 440 terabytes storage. And just to give you an idea of what that's like, a typical desktop computer might have something like 4 cores, 48 gigabytes RAM. Note we have terabytes, and maybe 1 terabyte of storage. This is what you might typically expect from your own desktop or laptop computer at home. So what we have is really much more powerful. The picture on the right is not a picture exactly of our servers, but it's that those are what servers look like in our own, and we do have a server farm that looks similar to that. Uh, so we use those servers as well as their expertise to provide three main services to the research community. We provide direct access, which is when we give people login credentials, an account with our servers, so they could access our service remotely to do their own research. Sometimes they lack the expertise or experience or time to do their own bioinformatics analysis, so they come to us and commission us to do it for them. So a custom analysis to meet the needs of their particular research project. And then we also undergo a training. We have different training programs, internships, uh, training seminars or modules. We have immersion programs for people to come here physically or now remotely to engage in a longer form project under supervision, all under the rubric of bioinformatics training to help people enter into or gain experience in this field. Uh, so when it comes to direct access, clients are allowed to, like I said, access in our servers and run the programs that we have installed in our servers. We have many uh, bioinformatics software available, and many more can be downloaded for free and installed by request if that's what they need. And we also have local copies of major bioinformatics or sequence databases, such as the NCBI NRNT. That's what's used when you do a basic blast search, if you're familiar with that, for example. We typically use free and open source bioinformatics software rather than proprietary or commercial software to ease the path to availability, not just for ourselves, of course, but for the broader research community. Because so much of what we can do in bioinformatics, you can do with free and open source software. You just need the know-how to use it. And then with custom analysis, like I said, we can do bioinformatics on behalf of the client. So we work with them to put together a sort of plan or workflow that will address their specific research questions. They provide us the data that they have, and then we work with them in implementing that workflow, scripting it, running the programs, running them on their servers, because a lot of things a lot of bioinformatics analysis you can't just run on a typical desktop computer. It would take years and years now, so we have to run it in our servers, and then we give them a final report on our findings. And of course, we have our trainings. We've had many of these over the years. I miss the time when our trainings were more in-house now because we have, and you can see pictures here of our training rooms. We've had internship programs, the immersion program, like I said before. Sometimes thesis students come to us for supervision, and we work with both the thesis student and their principal investigators to help supervise what they do. Now, because of COVID and all of the constraints that's imposed, our trainings are less frequent, and when we do have them, they're remote. But I hope that we can come back to something like this soon. Uh, so I keep talking about bioinformatics now, which might not be clear to many if you have a more classic biological uh, background or training. What precisely consists of bioinformatics may not be clear. Uh, so I want to give here the basically the textbook or standard definitions. I'm giving the definitions for both bioinformatics and computational biology, which tend to be sometimes used interchangeably, but at least the NIH working group came up with somewhat different definitions, where bioinformatics uh, is more focused on the development and application of tools for expanding the use of biological data like sequences, including specifically addressing aspects such as the acquisition, the storage, the organization, and analysis of such data. Whereas they consider computational biology to be more of, whereas bioinformatics is a little bit more like engineering in that it's building and using tools, computational biology is more of the science, which is more about the development of theory, of methods, of models, of simulation techniques in order to address research questions in biology. So technically speaking, that's sort of where these two terms differ. But practically speaking, feel free to use them interchangeably, as most people do. Actually, you can go even simpler than that, no? Essentially, bioinformatics or computational biology is simply the merging of multiple disciplines, the merging of biology and the biological sciences with other computational disciplines such as mathematics, computer science, and statistics in order to address research questions in biology and medicine. So, which is why it can be usually presented as something like a Venn diagram, where the union of all of those things together can be considered as bioinformatics. But actually, I like this figure more, as it speaks not just to the multidisciplinary nature of bioinformatics, the interdisciplinary aspect of it, but also somewhat the chaotic hodgepodge of the way all these disciplines come together and how many different people from different institutes, different disciplines, different fields of expertise come together to address these problems. Some of these are terms you might be very familiar with, and some might be completely new or different to you, depending on what kind of background you come from. So I like this as a sort of visual representation of what bioinformatics work or research is really like. Uh, so, while my talk is ostensibly about bioinformatics as a whole, of course, being part of the Philippine Genome Center, my main focus is on computational genomics or computational genetics. These days, those two terms are also somewhat interchangeable. Nano. But when I talk about computational genetics or genomics, just think of it as bioinformatics, basically, which is, of course, the use of computation and statistical analysis to decipher bio, uh, the to decipher questions in biology from genome sequences and related data. So those are, of course, the actual DNA and RNA sequences that we develop from high throughput sequencing, uh, protein sequences and structures. Then there's gene expression data, microarray data, methylation data, and of course, you combine that with the phenotypes of interest in our research, population level data, other metadata that might be incorporated or correlated with the genetic data we have on hand. Uh, So in computational genetics or genomics, we're concerned with research questions as, for example, what is the size or structure of a particular genome? That might be one of the first questions you'd ask. If you want to look at the genome of a particular and poorly understood species, that's what you'd ask. So, gaano kalaki yung genome na to? And once you've looked at the basic size and structure of the genome, of course, you'll ask next, what's inside of it? What are the genes and regulatory elements contained in it? How is it organized? Is there a lot of duplication? Is there a lot of junk DNA? What is the density of the genes involved? How are they distributed? Uh, is there a lot of duplication and so on, recombination? And then you might want to compare the genomes of different individuals or species. This falls into the field of comparative genomics because that can give clues as to how the genetic differences might or genomic differences might map to the phenotypic differences between individuals or species. And that gives us clue as to how a phenotype can arise from genotype. And of course, ultimately, the questions we are trying to address are how does all of this information, this genetic and genomic information, relate to an organism's biology? Because, of course, in a large sense, that is what genetics and genomics is trying to entangle the very complicated webs that tie together the DNA sequences and RNA sequences in organism's genome with its actual physical structure, its phenotype. Uh, so these are, of course, the very fundamental questions we want to address in genomics. But there are also many other related fields. For example, transcriptomics, which is concerned with, as the term suggests, RNA transcripts that might be present at the sample. We could ask ourselves, what are the levels of expression uh, for various RNA transcripts, and are they associated with what biological structures or environments? What biological functions or molecular processes are represented by these RNA transcripts? And now this is vary depending on anything, depending on the organism, on the tissue type, on the cell type, on the stage of development, on the environmental conditions, on on the treatment, on the species, on whatever. Because of course, the way that the what's dynamic about a living, breathing organism isn't the genome, which is barring parang somatic mutations fairly static, but rather the transcriptome, no? Because different genes get expressed at different times. And we want to understand why that is and how that relates to the organism's biology. And then there is the field of metagenomics, uh, which relates to assessing the genetic composition in a mixed environmental sample, typically an environmental sample comprising of microbial species. You want to, let's say, you take a sample in, I don't know, soil or seawater, and then you sequence what you can inside that mixed sample rather than one that's purified, and you get clues as to what is the microbiota living in that sample, what are the microorganisms living there, and at what relative levels of abundance, what are the functions expressed by the genes within that community, how does that species diversity and community composition in that sample vary across space and time. Maybe a soil sample in one location has this microbial community, and the soil sample in this just 1 kilometer away has this microbial community. What explains the difference? How are they different, and what would explain those differences? How would that correlate to other factors in the environment, for example? So those are might be questions that are addressed in metagenomics. And it barely scratches the surface. Now it's trendy to name fields by omics, no? Under the broadest field of parang genomics, parang ngayon pro-omics na. Uh, Proteomics, epigenomics, metabolomics, exomics, and well, there's the outlier systems biology. And I really don't have the time to go into detail into any of these, but I hope you can appreciate just how vast these emerging fields are even today. Uh, and maybe you'll be interested in focusing on one of these in your future careers. Of course, with all of these research questions and objectives, there are multiple challenges involved in these fields, no, in bioinformatics and computational biology. One, perhaps the biggest and most pervasive one is scale. The reason that genomics exploded as a field in these last couple of decades is that next generation sequencing, the technologies involved, have given rise to massive explosion in the available information to process and analyze. Uh, a lot of you may be too young to really remember the heyday of the Human Genome Project. This was a massive collaboration among multiple countries and research institutions to develop the first full sequence of the human genome. And it took something like 13 years officially from the initial founding to the announcement of its completion, and something like close to $3 billion to produce one genome. Now, a human genome could be sequenced in at most a week, and that's with all of the library preparation, the actual sequencing run, the analysis afterwards, and it takes under $1,000. From $3 billion to 1,000. Actually, 1,000 is a lot. Uh, the A lot of places can do it for a lot less, and I think there's a firm in China that's claiming that they can or will be able to do it for $100. So that's again an order of magnitude less. So the costs and time involved in sequencing a genome has vastly uh, gone down compared to just a decade, 2 decades ago. And of course, as a natural consequence, the number of genomes that have been sequenced, not just human genomes, but genomes that just about every species scientists can get their hands on, has exploded. Please note here that the cost in the plot for the cost in genomes, this is plotted on a logarithmic scale, hindi yan linear. Kaya ang baba-baba na ngayon. And this is parang the overwhelming fact that faces basically all of modern genomics. In a way, practically all of biology. I don't think there's a field of biology that hasn't been somehow impacted by this. Even fields like ecology, cell biology, and so on. Lahat yan impacted by next-gen sequencing technology. Excuse me. And then, of course, once you've sequenced the genomes, there's the challenge of assembling and annotating the genomes. Assembling a genome, well, what comes out of a sequencing machine are very short fragments of the original input sequence. They're called reads, and it's like you shredded the newspaper. You have to put it all back together. So that's assembly in genomics. And then once you put it back together, you have to read it and figure out what's inside it. What is this genome telling us? So that's a problem of annotation. And I'm really just including these two here, assembly and annotation, as a sort of stand-in or proxy for all of the other related downstream analyses that might be involved once you've done the beginning work. Because beginning work pa yan, assembling and annotation of a genome. And then, of course, if you're not a, if you come more from the biology side of things rather than the computational side of things, you may not really think of this too much. But storage and compression is also a challenge when it comes to the explosive growth of sequence data. Sa amin, there's always a need for us to try to find funding to get more servers, for example, both storage and compute servers, because the genomic data just keeps piling up, and this isn't, of course, just a budget problem in terms of buying servers and buying the maintaining the storage space you need to store those servers, but also a computational problem. You need things like compression algorithms and so on to be able to efficiently archive and access all of this complicated data. A related problem is access and privacy. This is especially relevant with human genomic data and medical data. where of course we have to balance the scientific and research needs of giving access to the people who need it so that they can address the research questions, with of course protecting the private information of patients. Uh, so, and this can be a concern not just with facilities like us or researchers like you, or what you will be someday, but if you've heard of parang things like ancestry.com or MyHeritage, where you parang give them your genomic data and then they'll tell you where your ancestry comes from. One of them, I think it was MyHeritage, actually got hacked a few years back, exposing a lot of genetic information. And it's tricky here, no, because you don't even have to share your own information for you to be identifiable. People have actually caught criminals not because of DNA they've left at the scene, but because of DNA that their relatives have submitted to these kinds of databases. So this is an ongoing challenge, and not just a technological challenge or research challenge, but also an ethical challenge, where we have to address all of these parang competing needs, research and privacy. Uh, and then there's the problem of managing that information in databases. A database is not merely a warehouse where data is stored. It has to be functional. It has to be organized. It has to be accessible. It has to meet the specific research needs of whoever is using that database. And every researcher has different needs, so it has to be flexible. But that flexibility itself introduces its own cost in terms of maintaining it, in terms of parang developing the schema for the database. So this in itself is a parang a complicated problem. Again, I imagine most of the people I'm speaking to have more of a biological background than computational. So maybe you're not too aware of these things, but there's a lot of thought that goes into a lot of the debate databases you might commonly use in, for example, NCBI. Parang ganyan yan. Uh, and then, once you have and can manage and access all of that data, you have to be able to uh, formalize it in models that are useful, that are sufficiently accurate. No model is, of course, perfectly accurate, but one that captures the essential features of the biological phenomenon you're trying to address, something that's tractable, something, and these models can help us develop the theoretical foundations of whatever we're studying, and generate and test hypotheses. A big part of science is really making and developing and testing models. No, we don't always think of science as modeling, but that's really what it is, whether it's a structural model, a formalized mathematical model, an animal model. A lot of this is we're trying to understand reality through models. And now bioinformatics and computational biology gives us a challenge to produce mathematical and statistical and computational models to help us understand very complex phenomena, and to be able to summarize all of those phenomena in terms of basic terms and principles that we can understand and test. So it's with all of that that I like to say that all biology these days is computational biology. And I quote a specific paper for that. It's kind of true, no? Um, it's kind of an uncomfortable thought to many biologists, maybe, and maybe aspiring biologists. I'm sure a lot of the people that I'm speaking to are interested in science, but went into biology because you don't like math. Admit it, it's true. A lot of biologists are not the people that are parang uh, particularly inclined towards the mathematical fields. But today, well, the truth is that all biology today intersects or connects with the computational disciplines, because the amount of data that we have now could not be made sense of, could not even really be stored or accessed with the classical or traditional approaches. There's so much data now, and more is coming in, that we need to be able to organize and address all of that through the tools of computational biology and bioinformatics. Fortunately, just because you're a biologist doesn't mean you have to be a computational biologist. It means you just have to hire one in your team or collaborate with one and know how to speak to them. So yeah, we all have our own disciplines and expertise. Uh, but computational biology is just one and one that's growing in importance today. So what can computational genomics and bioinformatics do? Uh, so much. No, what can't it do might be the easier question. Uh, if you're interested in going into sciences to help the country, help humankind, as I hope you're inclined to do, there's no shortage of applications in bioinformatics and computational genomics that can help you do that, no? Because like I said, if all biology is computational biology, then anything in biology that helps people, computational biology can contribute to. For example, in facilitating conservation efforts, in the discovery of novel natural products, uh, in conservation efforts, it's not just field work. It's not just protecting endangered species out in the field or out in the wild. It's also making sure that wild populations, even captive populations, have a healthy amount of genetic diversity. And to do that, we need to understand the processes that govern genetic diversity within populations or across species. And to do that, we assay genetic markers or we sequence their genomes, and then we utilize different fields in population genetics and molecular phylogenetics and bioinformatics in order examine how uh, to examine that diversity and see how that diversity is interacting with various environmental processes, various population processes, uh, and that can guide efforts in conserving endangered species. Or if you want to, excuse me, if you want to mine the natural world for natural products that could be of use in pharmacology or medicine, that computational genomics and related fields can also help there. The traditional approach is extracting it's largely in the field of chemistry and biochemistry, no? You extract all of these compounds in whatever plants or corals or whatever you have. You assay them for whatever uh, effects you might think they have, and then do further experimental work. And that work is ongoing. But now that can be informed by computational techniques such as synthetic biology or parang structural prediction, basically piping genetic and structural data through computational workflows that can help to streamline testing of hypotheses, streamline the generation of hypotheses that you can later test with experimentation. So it can help you focus your efforts and the resources so you don't have to assay literally every compound you were able to extract from this whatever plant in the rainforest, but rather maybe narrow it down to specific candidates that you think does what you want it to do. Uh, Computational genomics and bioinformatics can also facilitate breeding efforts and improve crop yields. Traditional approaches to animal husbandry and crop breeding is basically you have the organisms, plants or pigs or livestock or whatever that have the features, the phenotypes that are of interest. You make them breed, and then you look at their offspring. You find the ones that all inherited the features that you want. You you crossbreed with others that also have the same phenotypes, and then so on. So you basically have to wait for crops or animals to grow up to adulthood so that they can be their phenotypes can be properly assessed, and so that they could be uh, included in further breeding or animal husbandry efforts. But now, with computational techniques and genetic information, we can have more refined predictive models and also find the connection between marker data, genetic markers, SNP data, the genotypes, to the phenotypes of interest. So when you've bred the plants or animals, you can look at their genomes and then find and immediately figure out, these specific individuals have the markers that I need, and I know the correlation of that marker with my phenotype, and that can be included into my model, so you can have more refined prediction of what will happen if you breed this strain with that strain, that breed with that breed, and this makes it easier and more efficient to more quickly produce the strains or crops or breeds that are of agricultural or industrial interest. Uh, these bioinformatics can also reveal mechanisms of drug resistance in pathogenic microorganisms. Unfortunately, this is kind of uh, uh, parang resistance of microorganisms is kind of relevant today, sadly. Uh, but yeah, um, this is another example of how uh, these disciplines can help to correlate a genotype to phenotype, except the phenotype of interest is drug resistance or uh, antibiotic resistance, which is important in addressing uh, problems in medicine. You can use bioinformatic analysis and computational modeling to see how not just a microbe, but maybe a microbial community is evolving, not just in a controlled parang lab experiment like in a petri dish or in a test tube, but maybe in an environmental sample, maybe in a gut microbiome, in a tissue sample, and then combine the different marker information that you have with the different treatment environmental information available to you to be able to figure out the underlying mechanisms involved that's governing the evolution of those microorganisms. Uh, Computational genomics can also help to determine an individual's susceptibility to disease. Uh, she, Angelina Jolie kind of became the sort of like the poster child, or at least the most famous case of this. No, she very famously had a double mastectomy because genetic testing revealed in her a genetic predisposition to breast cancer. So this is another example of how, excuse me, uh, how genetic information and the models and analysis involved are able to help us correlate what what variants a person might have in their own genome with factors that are of immediate medical relevance, such as susceptibility to disease. Not just necessarily cancer, uh, but really any hereditary disease or any disease with a hereditary component, of which there are many, from Alzheimer's to alcoholism to really any number of disorders, and even things that indirectly would affect their susceptibility to communicable diseases because variants that might affect the health of their immune systems, for example. So I wanted to end my examples of the applications of bioinformatics with examples related to health because I want to close this talk by talking a little bit about our genomic biosurveillance program, which is a partnership between the Philippine Genome Center and the Interagency Task Force on Emerging Infectious Diseases, the Department of Health, and of course, other testing labs throughout the country from which our samples are derived. Uh, so, the objectives of this project are to monitor and identify the occurrence of new variants of COVID-19 in the country. This is what's been in the news on occasion these last few months. examine the spread and prevalence in the various regions in the archipelago, which can help uh, government and health officials to direct resources. And we also want to use all of the genomic information to provide policy recommendations to the IATF on the appropriate response regarding the variants that we've detected. So the way this works, I'm just going to give a very broad overview of our basic methodology uh, with guidance from the Epidemiology Bureau of the Department of Health. Samples are collected from COVID-19 positive individuals across the country, from partner hospitals, institutions, testing labs, clinics. Those samples are sent to us for RNA extraction and then cDNA synthesis and genome enrichment. The RNA extraction is performed by the Clinical Genomics Laboratory here at PGC. And then our DNA sequencing core facility is what does the cDNA synthesis and enrichment. And then, of course, the sequencing core facility does the actual sequencing of the cDNA using an Illumina COVID-seq workflow. We try to target 750 samples weekly, but due to various constraints, sometimes we do more, sometimes we do less. Excuse me. And then it's passed on to us at the PGC core facility for bioinformatics, where we do the analysis that is then sent to our partners and to the government and to guide their efforts now. So what, like I said earlier, what we get from the sequencers are sequencing reads, which are then mapped to a reference genome, the reference SARS-CoV-2 genome that the entire global scientific community is using. And then we produce an assembly. So every sample, we determine the genome sequence of that sample. And then we look at the variants. We look at the each sample for what mutations are present in that sample, because of course the virus is naturally evolving, and every time you sequence a sample of SARS-CoV-2, it's going to have its own specific pattern of mutations. And then we do various downstream analyses. One of them is we compare them to each other and to what's already been released and publicized elsewhere, and to identify the specific variant or lineage to which it belongs. We do other analyses that might be of relevance to its epidemiology, like phylogenetic analysis and haplotype network analysis. And then we tally the mutations that we've seen in mutation profiling to see if any new variants are emerging. Uh, and this is the kind of work that led us to the detection of a novel SARS-CoV-2 variant in Central Visayas. I imagine that many of you might have already heard of this. This has been in the news for some months already. We noticed a particular pattern, a distinctive pattern or profile of mutations in SARS-CoV-2 samples collected in Central Visayas that wasn't quite like the other uh, variants that we've detected before or that are already available online now in public databases. And disturbingly, it had a large number of mutations that were already flagged as mutations of concern in other variants of interest in other countries or regions, such as the UK and South Africa. Uh, and then we also did phylogenetic analysis, which confirmed that it does indeed appear to be a novel variant. Ito siya. Now it's been officially designated as P.3 here, and it stems from the larger B.1.1.28 lineage of SARS-CoV-2. In fact, it's actually already been detected in other countries, including the UK, Australia, and Japan. Uh, and in addition, of course, all of these variants that we detected and the others, the UK variant, I should refer to them by their technical names now, B.1.1.7, first detected in the UK, B.1.351, first detected in South Africa, and then the P.3 lineage that was first detected here are the ones that appear to be most prevalent in the Philippines. And these are what we've been reporting to the Department of Health and the IATF for them to guide their efforts and allocate resources in controlling this pandemic. I'd like to apologize a little bit. I'd like to go into more detail into this work that we're doing, but uh, like I said earlier, no, privacy and management of data access are important. I'm not cleared to talk about a lot of our immediate results, so I can only speak about what's already been made public. So this, for example, is a copy of one of the, I think, the most recent release by the Department of Health on what's been detected in our most recent sequencing batch. So this is work that is important, and on a personal level, I find to be meaningful. No, it's when you work in sciences, sometimes especially in the lab, sometimes the how it can translate to helping other people isn't easy to see. You're just in the lab, parang running experiments with your pipette or whatever, and you write papers and you parang submit findings, you present it at the conference, but it's not always easy to see how that really directly helps other people. So it's kind of gratifying, but also very scary to see how the work that we're doing now translates so quickly to policy and to parang news of concern to the country and to the world. So on a personal level, it's very fulfilling to me from in my career, but also because of the stakes involved, it's very overwhelming and scary. But I don't think there's anything else I'd rather be doing right now, but this sort of work, given the state that our country and the world is in. With that, I'd like to acknowledge, in addition to my immediate core team members that I mentioned earlier, also the other people that we've hired on to help to tackle this project with us. Ariel is our one of our staff in IT, and then we have our researchers, Kenneth Kim, Mark Castro, Rihanna Cruz, Ria Yangzon, Aaron Pangalinan, Rija Mantaring, so I want to thank all of them for the incredible work that they've been doing these past months in this biosurveillance project. And I think I would like to close with that. Once more, I want to give a sincere thanks to my hosts for inviting me and giving me a chance to speak on this topic. And I want to thank all of you for taking this time to listen to this talk. Thank you very much. And that concludes my talk.
Thank you for that very bioinformative lecture, Sir Lapid. The floor is now open for your questions on the discussion of Sir Lapid. For the attendees in the Zoom meeting, you may key in your questions through the meeting's chat box. And for our Facebook Live attendees, you may comment your questions in the comment section. These questions will be noted by the organization and will be addressed on our post-webinar publication.
We have a question from Ms. Princess De Rosa. Given the advancement of the genetics and molecular biology field, such as the discovery and application of CRISPR or Cas9 technology and gene therapy, which are greatly accompanied by bioinformatics, how confident are we that we can apply these to next generation analysis and approaches in our country?
Okay, that's a great question. Huh? It is very broad. I'm going to specifically address the example in the question in CRISPR-Cas9 technology, which, if I'm being honest, is kind of outside my expertise. I've never directly handled any project relating to that. Of course, I'm familiar. Uh, as I understand it, CRISPR-Cas9 technology is already used in many laboratories uh, around the world, including the Philippines, if I'm not mistaken, uh, as a useful tool in analyzing. Um, well, I think it's largely used for mga animal models, no. Um, as far as gene therapy, that raises a lot of ethical uh, issues and questions about parang changing the human genome, which is, well, something that isn't to be entered lightly. I am confident that these are very useful and powerful tools that are and will continue to be utilized in the advancement of genomic research. Uh, and I have trust that these technologies can and will be used responsibly. Uh, yes, I hope that answers that question. I'm afraid I can't give too detailed an answer. CRISPR-Cas9 isn't something I'm very deeply, intimately familiar with. I mean, in terms of what's currently going on.
Our next question is from Luis Brian B. Renera. His question is, these days, more people start wanting to know about their DNA. Do you think there could be a time in the Philippines where people could just upload their genetic profiles to learn about their predisposition to certain diseases or search for long-lost relatives?
That's interesting. Uh, hmm. Um, in a sense, I mean, this is already available now. I think the question emphasized in the Philippines because we know that these sorts of services are already commercially available in other countries, like the US. And actually, I don't think anything's stopping a Filipino from submitting, using the same services, except maybe the mailing cost of shipping their samples overseas. Uh, I want to caution uh, the it's not so simple. Okay, to directly address the question, the Philippines isn't ready to do something like this yet right away. I mean, yes, you can already use what's already commercially available out there, but predisposition to certain diseases, for example, is something uh that is more complex because these parang predictive tools are population based. You get you assay the amount of diversity in a specific population. For example, let's say the American population, and then you look for correlations with diseases, find markers, and so on. But an association with a marker in a specific population might have a different association in a different population. So you need to have a lot of comprehensive parang analysis in your specific target population for tools like these to be useful. So if you submit your sample to one of the American or European based services for things like this, and it tells you that you have some predisposition to diabetes or whatever, you can't fully trust it because that prediction is based on genetic information from a different population country. So I think it might still be a while before that sort of work is done here. We're still, even now, barely beginning to scratch the surface of the amount of genomic diversity we have in people here in the Philippines. There's already been a lot of work in animals and plants and so on, but because of the greater ethical challenges involved, um, the the medical issues, the concerns with privacy, with storage and access, we're still very much at the beginning of doing that kind of work here. So I think, of course, there will be a time when we'd be able to do that, but it's not right around the corner. Is what I'm saying. I hope that answers your question. Maybe that's a little bit too detailed, but yeah.
And thank you, sir, and thank you, Brian, for giving that question. For the next question, we have John David Cortez. What is the general landscape of bioinformatics in the Philippines with respect to the number of active practitioners and the institutions that are able to practice or develop the discipline?
Oh wow, that is an excellent question. And hmm, this is a question that directly pertains to the work we have at the Philippine Genome Center. Specifically because one of our mandates is to elevate and uplift that number of active practitioners. Now, a lot of the people that do bioinformatics out there are people that we've trained or that have at least attended talks or seminars with us or that come to us for trainings. Like I said earlier, our trainings have kind of slowed to a crawl during the pandemic. Uh, but we still continue to get requests for training, for seminars, because people need that. Like I said, biology is computational biology. In terms of what the general landscape is like in the Philippines, I'd say that the numbers are growing. People need that. Like I said, biology is computational biology. In terms of what the general landscape is like in the Philippines, I'd say that the numbers are growing. Uh, the Philippine, I wasn't able to mention it earlier, but the Philippine Genome Center has satellite facilities in Visayas and Mindanao, both of which have their bioinformatics teams that we consulted with us and that we helped to set up and that we work with and that we collaborate with many other people and many other people that we haven't trained utilize our resources. So there are many there are well I want to say hundreds I guess of bioinformatics in the Philippines, but the true and the most honest answer to that question is there aren't enough. We need more people that are experienced in these fields, uh, that have the training. It's a challenge because bioinformatics is multidisciplinary. You graduate with a bachelor of science in biology. That by itself is not enough, because then you still need some basic understanding, at least of the math, of the statistics, of the programming, of the computer science, and so on. If you don't have to do it yourself, at least know enough to be able to collaborate with or work with someone that does. So the barriers to entry into bioinformatics are in one sense pretty high, in that you have to at least have a basic familiarity of multiple fields. But as an encouragement, especially to those of you that are coming to the biological sciences, some of the barriers are actually very low. Because if you feel like, pero hindi ako marunong mag-program, hindi ko kaya yan. If you want to learn to program, you can learn that for free. It's easy to, there are so many online resources for you to learn tools like R or Python, which are basic skills that you need to get into doing bioinformatics research later on. So yeah, it's a complicated question. It's hard to generalize what the landscape is in the entire Philippines in just a short answer. But if I did have to give an answer, I'd say that landscape is still too small, and we're doing our best to expand it further.
So thank you so much, Sir Lapid, for that answer. Due to time constraints, we're only be going to accepting three more questions after this. So we have another question from Ms. Frances Alexandra Isberto. Questions regarding the mortality of some scientific processes have always existed, such in the case of GMOs, CRISPR, etc. So thank you so much, Sir Lapid, for that answer. Due to time constraints, we're only be going to accepting three more questions after this. So we have another question from Ms. Frances Alexandra Isberto. Questions regarding the mortality of some scientific processes have always existed, such in the case of GMOs, CRISPR, etc. Do you think this kind of questions will also be brought up in the advancement of bioinformatics here in the Philippines, given our very conservative culture?
Oh wow, that's a very heavy question. Huh? Um, the ethical questions involved, uh, at least with the specific things you bring up, GMOs, CRISPR, and so on, are a little bit outside of bioinformatics. Not to say that bioinformatics doesn't address its own ethical questions, especially when it comes to things like privacy and access and ownership of data. But when it comes to things like GMOs or by CRISPR, I imagine that relates to things like genetic modification. These are questions really better addressed by doctors, by ethicists, by researchers in those fields. Bioinformatics and computational biology are pervasive, but not so pervasive that all the answers come from us bioinformaticians. No, you say na but your question also addressed the conservative culture of the Philippines. That again is I'm not even sure that I'm qualified to give that answer. I'm not a sociologist, but I am optimistic. On a personal level, I am optimistic that uh, that the needs, the dire and the vast needs of our country will inspire people to address these questions as needed, because we can't just not use technologies that can help people, right? We can't just ignore them. We have to be able to use them in the responsible and ethical ways. To what extent bioinformatics can help, no? In that bioinformatics as a computational science can address hypotheses relating to risk or harm. Uh, because risk and harm are scientific questions. And by developing empirical answers to those questions with the tools we have available, we could help to guide policy and maybe convince people of what is and isn't safe or appropriate to use. That's my answer. Thank you for that very challenging and insightful question.
Thank you, sir, and thank you, Francis Alexander, for that question. For another question, po, we have um, from a question from Armil Pequeña. With the current state of bioinformatics in the Philippines, how will the current or potential bioinformatics approaches or strategies help address national health issues and further uplift the public health in the country?
Oh, wow, that is another very big question. Well, I've already spoken a bit about our biosurveillance project, right? Excuse me. Which is one way that we do our part to address questions relating to the COVID-19 pandemic. Another is a project that we've recently gotten funded for and launched is the Philippine Virome project, uh, which involves developing a database and analysis tools for the storage and analysis of virological data. Very relevant now because of COVID-19 again, but this is still a separate but related project, um, which involves assaying, looking at the diversity and variants and phenotypes of the viruses that can be isolated and extracted in this country, which hopefully could give us the tools and information we need to maybe prevent a future pandemic, or at least address or prevent future epidemics or outbreaks, or help to develop maybe cures or vaccines. And that's just one example. The truth is, bioinformatics and related fields like medical informatics already pervade much of the medical and biomedical sciences. No? Um, just things like medical informatics is critical now in managing all of the patient health information across different institutions, for example, and combining that with genetic data wherever it's available can help us to address medical questions even more targeted ways, as I mentioned earlier. But we do still have that, I don't want to say bottleneck, but that parang obstacle, that challenge of uh, the the greater hurdles involved with collecting human genomic data, because there are many important ethical and security and privacy issues involved there, which should be responsibly addressed. I'm not saying that there are things we have to overcome na dapat wala na mga ganung privacy issues or we should ignore them. Absolutely not. As the responsible thing as scientists is to be able to safeguard that information in proper ways. But that does mean that there's relatively, to my knowledge, and my knowledge is not all encompassing. I'm not a doctor, for example. To my knowledge, the collection of human genomic data is still limited. But pathogen data is less limited. Of course, there are less, we don't need consent from viruses and bacteria. So that's advancing at a greater rate. Hopefully we could advance more quickly on that front, if not the human genetic. Yes, thank you also for that question.
Thank you so much, Mr. Lapid. So due to time constraints, we will be ending the question and answer here. So for additional questions, for other queries from our audience, kindly key in your questions on Facebook Live comment sections so that we can compile and send these queries to our speakers, and answers will be posted in our post-webinar publication. Once again, we want to thank everyone who participated in our brief Q&A, and thank you for Sir Lapid for answering these queries.
Thank you very much. I really appreciate you having me here to speak about my work and answer these questions.
I am sure that everyone had their curiosity stimulated. If you want to learn more about bioinformatics, join us on May 1 for our second webinar, which will encompass advanced bioinformatics for research purposes.
SIHAY NBO is a national inter-high school biology congress organized by UP Cell Biological Society. This year's SIHAY zooms into the world of bioinformatics, decoding archives, unraveling the biological code through molecular data acquisition and analysis. With the advent of technological advancements that urge to unravel the closed-book science, bioinformatics proves to be a principal tool in collecting the essential codes to grant access towards the bigger world of biology. As we witness the hybridization of biology with other established fields in decoding the codes of life, let us look forward to bridging the gap between science and technology in evidence-based and precision studies through bioinformatics.
SIHAY NBO is comprised of several contests and sub-events, one of which is Bio-Symposia. Bio-Symposia aims to present recent advancements in biology and biological instruction to students and educators in the hope of using this knowledge to innovate their learning and teaching styles and methods for the advancement of biological education. Earlier, we discussed bioinformatics for the betterment of the Philippines with our esteemed speaker, Sir Carlo Lapid, a senior bioinformatics specialist at the Philippine Genome Center. There will be another webinar that will be broadcasted on May 1, having another esteemed speaker for the webinar. Are you guys curious about who it will be?
Got anything to share about the informative discussion of Sir Lapid? Or anything that you would like to share with your friends and relatives about this seminar? Use our official hashtags, #Bio-Symposia, #BeBioinformed.
We would of course like to acknowledge Sir Lapid for giving his time and effort and sharing his knowledge about bioinformatics for the purpose of this webinar. The UP Cell Biological Society courteously presents this certificate to Sir Carlo Lapid. Allow me to read the citation. This certificate of appreciation is presented to Mr. Carlo M. Lapid, MSc, for his invaluable contribution as a speaker in Bio-Symposia, Bioinformatics for Novice Scientists, as part of SIHAY National Biology Olympiad 2021, given this 25th of April 2021. Signed by our President, Bruce John S. Castillo, and our Educational Committee Head, Princess Amy R. De Losa. We would also like to invite our attendees and viewers to please like, share, and follow UP Cell Biological Society's official social media accounts on Facebook at UP Cells, Twitter at UP Cells, and Instagram at the UP Cells. You may also want to follow us on our SIHAY NBO Facebook and Twitter page at SIHAY NBO to keep yourselves updated on the National Biology Olympiad.
Before we call it a day, we would like to prompt everyone that we will be having another Bio-Symposia on May 1, Saturday, from 2:30 to 4 in the afternoon. Finally, we would like to once again acknowledge our sponsors and partners. In cooperation with UPLB Computer Science Society,
UP League of Agricultural Biotechnology Students,
Don Honorio Ventura State University Biological Society,
Pampanga State Agricultural University Biological Society,
Biological Society Laguna State Polytechnic University Santa Cruz Campus,
PLM Society for Biological Sciences,
Bicol University Symbiosis,
League of Biology Enthusiasts, with Media Associates, UP Society of Management and Economics Students,
UP Biological Sciences Society,
Philippine Society of Cell Biology,
Biology Program Council. Special thanks to UPLB Development Communicator Society,
Symbiosis, the UPLB Biological Society,
and Agham Youth UP Diliman. For our attendees, please take note of the first code shown on your screen. The code is Decoding Archives, no space with letters D and A in uppercase letters. Again, the code is Decoding Archives, no space with letters D and A in uppercase letters, as shown on the screen. Participants are encouraged to attend the second webinar on May 1, as e-certificates will only be issued after the second webinar.
I genuinely hope that our attendees and viewers learned a lot from today's webinar about the field of bioinformatics.
We would like to express our sincere gratitude for all of you who joined us, the UP Cell Biological Society, in today's webinar.
Again, I'm Diane Shamka.
And I'm Elijah Rivera.
This is Bio-Symposia Bioinformatics for Novice Scientists. See you all on the second webinar on May 1. Thank you and have a great day.